we start? Sure. Okay.
Yeah,
Go ahead. All right. So I don't know. Do we announce the speaker?
Yeah, you announce.
Okay. Our speaker is Steve, who many, I think, most of us know, and will tell us about that title.
All right. It's just open problems, various things, as reverse order. So it starts with Smaldon. First with Smaldon, and then crowding, and then information transfer. So a little background here. I'll start with a quick Smaldon overview. I've sort of done that. Obviously, ask questions. Let's go along. This is all old stuff, but for all I know, maybe take the whole talk. We'll find out. Smaldon is a simulator which simulates particles diffusing in space, 2D or 3D space, or 1D space. Has particles, which are dots. They could be points. They can also have volume too if you want a volume. They can collide with walls, diffuse, can react to each other. That's a simple 2D picture. It's a 3D simulation of chemotaxis from Karen's work. We have E. coli cell, receptors, proteins in the cell, motor, flagella, motors. It's just playing some 3D geometry of the cell and diffusing proteins got to show that surfaces work. So molecules diffuse, best surfaces, and react. An example. This is the E. coli MIN system. So MIN system is a somewhat unimportant system for the cell. Helps find a cell division site, which is important, but it's been simulated over and over again by physicists, which I love the system because it's so beautiful. There's only a few proteins, MIN D, MIN C not shown, MIN E, also here in red. And the proteins cause this really amazing oscillation. So here it is in the biology. It says MIN D label, so I can forth in the cell. 20 second oscillation period. You saw here a small simulation showing got same simulation. This is all from published results. So simulation is new, but based on published model. So it's not red. Not for my side end of things. But sure, to start with well-mixed system, things pretty quickly go with this oscillation stage. So just here to show what Smaldon does. Particles diffusing, reacting, surface binding in this case.
So what is the function of this MIN oscillation again?
Helps us help in the cell center. So the idea is that MIN D is at both ends alternately, but not in the cell center on average. And so MIN D inhibits the center ring formation. That's because that's the center, ring is the center. Yes, it's a really beautiful system for physics. Smaldon does single particle tracking, for example. You're looking at, say, like actin filaments on a surface of a cell membrane, so it's a 2D system in this case, and watch single molecules fusing around. And they get sort of stuck in these corrals for a while, then just hop across the filament to the next corral.
What rule did you use for distributing the actin filaments randomly?
I picked random endpoints and connected them to filaments.
Uniformly?
I think so. Okay. I said pick 100 pairs of endpoints, each one random, draw lines. So.
And what is the rule? So, but when they are on the filament, do they move up or down, or they just?
They're not filament bound. They're here. There's a molecule between filaments. There's a cell membrane. Here's the idea. The membrane is underlain by filaments, and so it's diffused in the cell membrane. They can hop across the filament with low probability. So you have a lot of diffusion for a while, one corral, then hops over this corral, stays there for a while, hops this corral.
Okay, but I mean, so I might be missing something. So how do the filaments come into play? Because I mean, does it mean that? I mean, clearly the red dots are not diffusing freely, but what are you actually? How do sort of, in terms of mathematics, let's say, the filaments influence the diffusion?
Well, it's free diffusion within a corral.
They're like partially absorbing boundaries.
Exactly, partially absorbing boundaries, or partially transmitting boundaries, really.
Okay.
Yeah.
Thank you.
Yeah. Yes, this is actually probably to test out my partial transmission boundary equations. So hop diffusion, so things hop from corral to corral. Smolden uses. Smolden's actually been around for a while. It's been 15 years by now. It's been used kind of two classes of things overall for system modeling, for dendritic spines. Karen's work on hemataxis, E. coli min system. My work on yeast cell signaling. This is Alexander Jilkin's work. More yeast cell signaling, patch formation. Also systems here, that's my vision for Smolden. Also use a fair amount for biophysics modeling. Like here's crowding. How does, say, one molecule diffuse among a bunch of crowders? Or this is diffusion across a barrier, separates a dividing cell. This is Omer's work, there's no him on multi-stop phosphorylation. So pretty simple biophysics problems. It's not been used a whole lot compared to sort of BLAST or sort of big tools, but for this field, which is not a very, very small field, it's been used a fair amount. User feels Smolden. User writes a text input file. Here's predator prey with rabbits and foxes. And this is the whole file, it's pretty short and pretty simple to read. Puts that in Smolden, the black box. Out comes graphics, text output, then transports that into whatever you want. Excel, MATLAB, your own code. And
Sir, where do you describe the transport or motion in those input files? Are you going to talk about that?
Not really. Let's see, transport. So it's diffusion. Web-based emission, graphics OpenGL, 3 dimensions, these are other foxes, molecular per box.
So if for example, if the fox wanted to sort of do like a levy block or something. Sorry. So like the fox wanted to do something like go straight for a while and then diffuse or something, can you do that?
Not really. Things diffuse with plain standard diffusion. So Gaussian displacement is type stuff. Yeah, so diffusion coefficient here is 100 units. Also, yeah, I drift one. You gotta drift and say, always drift to the left at this rate plus diffusion.
You can't do that?
Yeah. So you're gonna drift.
Diffusion matrices. You can have more complicated diffusion matrices.
Yes, that's right. Also, and that's what diffusion too. You say diffuse preferentially on the x-axis, less than y, less than z. So, or off axis and a strong diffusion. I think I was never used my knowledge of this there.
Can your particles have orientations associated
No.
to them?
So they're dots with no rotations. The logic there is that rotational diffusion timescale is much faster than we care about here for the most part.
Swimming microorganisms, they're not, but okay.
Right. Yeah, for things like large clusters or not. But for most proteins we care about, they're really fast timescales.
What is this? It says reaction to rabbit plus fox gives fox plus fox 8,000. What's the 8,000?
That's the reaction rate constant.
I see.
These are all sort of arbitrary units. Because it's rabbits and foxes, not chemistry. It's in own internal units. But diffusion coefficient is 100 units. Reaction rabbit goes to 2 rabbits. Rate of 10. Rabbit plus fox goes to 2 foxes. Rate of 8,000.
And rate of 8,000 there means what exactly? That there's a probability per unit time when they're close?
If this were in real units, it would be, say, 8,000 cubic microns per millisecond. So convert that into per mole per second, and it's the same units roughly, or same.
You think of it as a well mixed rate, right? And
That's
you
right.
have to calibrate the
Yeah.
kind of parameters.
Yeah, so this is what the chemist says. The chemist says this reaction rate is this much per mole per second, 10 to the 5 per mole per second. They convert that into cubic microns per millisecond, which is a nice straight conversion. And then I put that into here, and it produces the same reaction rate at the end.
But how do you describe the locality of the reaction?
That's for starts out well mixed, then goes to steady state. So it goes to a steady state reaction rate.
This is spatial, right?
Yeah, it started out with a well mixed system.
Dave, you have a sharper question for me.
So a large box, well mixed A and B particles. And then reaction rate is a function of time. So here's a function of A molecules. Here's time. It goes down quickly. So it was. So that's 3 plus. So let's look at dA dt equals K of T. You'll see, starting well mixed, initially it's very, very fast reactions due to initially close reactants. That then levels off into a constant reaction rate.
What's your definition of close?
If you initially got an A here and then B right here, so it's with exceptionally close, you get that out of it. So very, very close. Those will react very, very quickly, immediately. And because of that, you have a very fast reaction rate. Later on, these close ones get used up. So what's left over are the sort of equilibrium distribution of molecules. Once equilibrium distribution is reached, then you have this constant reaction rate. So you approach a state where it's here's your pair distribution function. So initially, here's A molecule at position 0. Your B molecules are everywhere. So some are close, some are far, some are farther. However, things that are closer than this value, they react immediately. So these go away immediately. And you approach state where it becomes that shape. That's your steady state.
You're just trying to understand what he does in the simulation.
Okay.
I think they were asking like in the actual simulations, how do you handle biomolecular reactions?
Yeah, okay, we'll get there. Sorry.
Thanks.
Yeah, apparently off.
Well, this is how you're calibrating the
That's right.
parameters.
Yeah, so essentially the user says this is their value in real units per mole per second converted into volume per time. Small d is converted internally into internal parameters, which I get to. The reactions, rabbits, goes boxes, nothing. Start 1,000, well mixed evenly. Essentially, that's simulation actually. And here it is. What's this before? So here's, in this case, a 3D system. Spin around, it's prey or prey. Reds, rabbits, greens, foxes, and goes. So you can watch. If it were ODEs, get like a classic sort of sine waves, with PDEs, you got some sort of dynamics there. With particle-based, you get sort of boom-bust dynamics. So it's quantitatively quite different. It's kind of a little.
So, but there is no, I mean, so what is green and what is red here?
Green is foxes, reds, rabbits.
Okay, but it sort of seems that it's almost as where foxes are kind of moving where there are lots of rabbits in some sense.
Yes, once they get to them, because they reproduce basically,
Yeah.
you get more and more of them, and diffuses into that region. So there's no intentional diffusion, no intentional motion, just strict diffusion.
But it appears like, but that's what I'm saying. I would expect that, you know, given that your diffusion coefficient is constant, that
Right.
they would sort of be moving.
Well, it's due to this, more and more foxes develop. They develop at this front. So right here, for example, here's a little front. More and more foxes develop right there. Because of the develop there, spread from that spreading front, the front sort of moves in quickly.
Okay, so essentially, after the first time that sort of like the fox kind of meets the rabbits and sort of like that's why it's kind of it's not because there's preferential diffusion, but it's because when they're gonna meet the rabbits, they're gonna be eating. Rabbits are gonna be eaten quickly, and foxes would multiply.
That's right.
It appears like
Exactly.
okay.
It's just strict diffusion here, nothing special at all, but it appears to move in intentionally because of this building up, yeah.
Thank you.
All right, so this metaphor, small n is continuous space, not lattice, and finite time steps in 1, 2, 3D. So it's very simple in concept. You fuse molecules, do all surface interactions, all reactions, time step, time value, observe system, and repeat.
So surface interaction just means if I jumped out of my domain,
Exactly.
do some reflection or
Yeah.
something like that.
So diffusion,
Oh, sorry.
yeah, what this diffusion means diffuse Gaussian displacements. On surface diffusion, diffuse again to Gaussian displacement in 3D, then it's moved back to the surface by the local normal vector. So push there and vector. So that's all pretty simple, works well. Surface interactions, like I said, there's reflections off surfaces, absorption of surfaces, transmission through surfaces, adsorption, some sort of absorption probability or absorption rate, desorption with a rate, and partial transmission, some probability of reflections, probability of transmission. And also things on surfaces can hit more surfaces than the rest of those surfaces, so.
They can
wide
diffuse on the
interactions.
surface.
Exactly. So diffuse on surfaces and then more surfaces.
Surfaces have to be smooth.
The surfaces are composed of panels. So you build a surface and say, I want my E. coli cell to build out of 3 panels. I want a cylinder panel in the middle and 2 hemisphere end caps. Panels can be 6 shapes. There's triangles, rectangles, cylinders, spheres, hemispheres, and discs.
So you can have cusp, intersection of a ball with a plan, and you look at the intersection, you can
Yep.
have, okay. So you can handle reflections in this cusp, right?
Mostly.
Okay.
Yes, you can get, it's quite right. So. Yeah, things can get caught here. Reflections, it does multiple reflections in one time step, so things will say diffuse here to here. It says, okay, bounces off here, reflects that, oops, once again, off here, and reflect, reflect, reflect. And hopefully it comes out again.
Okay.
How
so
does
this-
it do the within time step correction? It's like when you overshoot to the boundary.
Yeah.
How are you doing the correction?
Reflects off with a sort of like a mirror, ballistic reflection.
You're assuming that it's moving in a straight line during
In a
the time
sense,
step?
not really. It's the correct probability outcome.
So you build a bridge.
Sorry?
You do a bridge?
No, it's a straight line reflection. So it says particle here goes to here, reflects off. So it's ballistic reflection off the surface.
Even though it's diffusing.
Correct. Because it's the correct probability distribution. So given a starting point, the correct distribution endpoint, this assembled correctly, despite the fact that it's not exceptionally lightweight. Word true diffusion is
Oh, sure, right.
endpoint result.
Yeah, but you can end up oscillating a lot here and wasting a lot of simulation time by,
In this thing here,
yeah.
but it's pretty rare though. And so what happens is you try to do it right, and then 50 tries, it can't do it, it says, oops, I give up. Puts back where it came from. That's very, very rare, but there's a sort of bailout approach there, so. I got stuck in this cusp, let's know, escape, try again. So mostly, virtually always, it works out great, and in multiple cases here, bounce back to space where it belongs, it's all good. Reactions, so this is the interesting part. A plus B goes to C, A, B diffuse, with one binding radius, then they react for a product. So the rule is within a binding radius, you always react. Outside there, never react.
So the rate then just determines the binding radius? Like that 8,000 number you put down?
Yeah.
I see.
Yeah.
So it's
the
not a
user-
rate in the sense of you have an exponential holding time?
Not at all.
I see.
In this case, well, the Stochastic formula for fusion limited reactions, K equals 4 pi sigma d. Sigma is the binding radius. If some steps were infinitesimal, you say, just solve this for sigma. Given user gives me K, user gives me diffusion coefficient, solve for sigma, that's my binding radius. That's a bit more complex than that because it's fixed time steps, not zero time steps, but that's it. Yes, that's reactions. These are more complicated, of course, with reversible reactions. Now, see diffuses for a while, then unbind, then dissociates. There's here I use an unbinding radius. So associates with the first reaction, unbinding radius here, sigma u. Reason, of course, is these two products could either recombine with a recollision or carry on and separate indefinitely. So hyperlimited radius gives a correct probability of recombination versus separating. This is a test of measure accuracy and likelihood. So here's first order reactions, which are pretty to simulate, and sure enough, it's not accurate. Solid theory, dots are simulations. Second order reactions, here's for a fusion limited system. The solid curve is Stochastic theory, dots are small and it lines up. And here's a noise picture, says noise is expected to get. So this kind of expected value here, and that's the noise envelope. And sure enough, it matches there too. So,
Sorry, in the previous slide where you had, when they have the disassociation,
yeah.
do you see there's a small probability to recombine with your parameter choices?
Depends.
So I guess my question is, what does it mean for the reaction? I mean, when people talk about chemical reaction rates for stuff to fall apart,
Mhm.
right? Now, how do sort of, events that go like fall apart, come back, and then separate, I mean, are they talking about the bare thing that's recombining, or is it sort of some coarser scale thing that's sort of the rate at which things permanently, or, on a longer time scale separate?
It depends on the measuring. If they're measuring actual, if it's like a fret measurement, a fret measurement.
Right.
And you're just measuring things pretty small distances, you can see where things are. But most often they measure equilibrium constants, nothing more than that.
Right.
They say on rate is make some value, and equilibrium constant is this value. Therefore, off rate must be this, is our net off rate.
And typically, what would be the range of probabilities to recombine that you see in the kind of applications you're doing?
So I use here a maximum probability of 0.2. So 100% probability of rebinding.
Okay, fine, that's
And that's plain, there's no data on that. They're published essentially. Maybe none. I just made it up. I said 0.2 seemed reasonable to me. So probably it diffuses apart forever, but it might not. If I use a smaller probability, then things get too far apart here. This seems unrealistic. It's higher probability, you're getting more repeat reactions, which seems wasteful. So I chose that number.
I guess you would see if when you did validations, just to like what the equilibrium ratios were, that would be off if this were a problem, right?
Correct. Yeah. This does give a correct equilibrium ratio. So it does give the correct overall rates and the correct equilibrium ratio.
Good.
Yeah. That's our key thing I aimed for. And the much more details on sort of the one molecule size scale, I feel, is a little bit iffy here because molecules aren't really spheres, they aren't really fusing this way. But what matters in the big picture, is that right? Yes. The last step here is observe and manipulate. For example, you can print out a number of molecules of kinds, or you can project molecules. This sort of is a really good sort of a hack part of Smallman. So the user says, I want this feature. Well, it's hard to add that to real code. Just toss it here, say, if user says this, then do it. Don't worry about why, just do it. Recently, Martin Robinson and with Radek's group, he and I added in hybrid simulation, so mostly Martin. This inner region here is a particle-based simulation with old-fashioned small bin. And then Martin wrote this lattice-based simulation with Mark Flegg's methods. Actually, basically less methods. Then Mark did the boundary between the lattice and the particle-based, and then implemented it. That's been a nice little feature to have in there. What's useful for is, for example, in this case, we try to have correct simulation in the small region middle, and then a good boundary to a effectively infinite space beyond that. I don't want to simulate a billion molecules, so I want to do that outside area of the core screen. So, nice little new feature. And then finish the little section. Right. New phone features. There's a whole bunch of stuff I'm working on right now. It's been in there for a year or two or three, and kind of gets wrapped up right now. And hopefully got finalized. Kind of boring, but install scripts for Mac and Windows. I could care less, but users do care, so yeah. Rule-based modeling, that's for complexes. So proteins aren't monomers usually, proteins are often complex into these large multivariate complexes. So how do you deal with the explosion of species and reactions? The answer is about NetGen, which is one written, to expand the whole list of species and reactions for me, import that into Smallden. That's essentially done. Accurate fusion sort of molecules, it's been pretty good within one panel, but crossing between different panels was incorrect before. And now it does it by saying, one panel, this molecule diffuses, say, here initially. Well, first thing to do is project it back to this panel. So move that thing down to that spot, and then bend it correctly around here. So up there. So now things will diffuse the correct fusion length across panel bends. There's also 3D as well. That's actually a huge challenge, but it should be easy if it wasn't. Live Smallden, I can call up Smallden from your own code, from C, MATLAB, Mathematica, whatever. Again, for trivial, form those input files. Before, input files had to have only numbers, and I can say, I want 5 plus 3. No big deal. So we're tracking. That's why it's pretty hard because molecules in Smallden, once they react, they go away. So how do I track a molecule as it binds, unbinds, unbinds, unbinds? Or track a single molecule out of a whole bunch of them. They can have a serial number and be maintained as it goes along. And last thing, white characters, she say, I want colors of all molecules that start with A to be green. So you say, color A star is green. So most of the stuff is not really interesting, but it's useful to have. Open questions, kind of what we're nominally here for. Filament simulation, discussed that last couple hours. Huge, huge topic. Lots been on there. Discussed before in this group, and it's a major goal. Moving surfaces, obviously a huge problem, huge task to do. And one of the things is adaptive mismatch time steps. Can you do time steps, either adapt and say, this is an intensive issue, let's slow down here and go more carefully, versus this is let's cruise along with big steps, or is mismatch saying, this molecule is pretty busy, let's do it in short time steps. Other molecules are doing a whole lot, let's do long time steps for them. So sort of a hybrid simulation, but within a single simulation, but different time steps for different things.
Are you parallelizing?
Yes, that's another goal. And that's a disaster. So parallelized at least 4 times. So first was MPI. It was all done in MPI, and the end result was slower than single thread. So parallelized next for P threads. Again, end result was slower than single thread. Parallelized twice for GPUs. Both of them gave 200-fold speed up and looked fantastic. But another one ran on my computer though, or the computer I could find. And so, yes, those authors claimed they gave great speed ups on their computers, but one portable to everybody else in all the world. So GPU is the way to go, but if I actually get it to work anywhere else, that'd be fantastic.
I've tried one of the versions that lost molecules and stuff, so I wasn't doing it correctly.
Yeah.
One of the GPU versions.
Plus, both of those GPU versions were written from scratch, and so they had no features besides kind of the core stuff. So if you want anything beyond that, start with GPU code. So yes, I'd love to have the core stuff in GPU, but it calls other features on CPU. So yeah. That's part one. Part two is crowding. It's actually using Smaldon. Well, as you know, cells are crowded. This picture from the big cell's work. This plasma cell is packed full of stuff. Actually, well, next slide says it. This work came from the Digital Conference, Russ Alradic and others, and has a group there to simulate crowding. We said, let's try crowding out in different simulators, in Smaldon, and special Gillespie simulators, and other things, and how it works with simulators. And that's really the goal of this project. There's going to be pretty interesting results, so I'm interested in them here. But for crowding, the basic picture is you have crowders, you have tracers, which diffuse among them. In this case, crowders are stationary, tracers are diffusing. In this case, tracers are playing dots. On very short time scales, or short distances, tracers are in free space and diffuse rapidly. So here are short time scales, diffusion is rapid. On longer time scales, start colliding into the crowders. On very long time scales, it's now long distance diffusion, and crowders sort of all average out to a uniform field. And so it's diffused normal up here and fast. Long time scale is normal, diffusion, and slow because of crowding. And in between times, you have this anomalous diffusion regime here. Diffusion kind of slowing down due to the collisions of crowders.
Is it being subdiffusing in that regime? Is that what you're saying?
So it's depends on the time scale.
Yeah.
Short time scales is normal, some diffusive at intermediate time scales, and normal and slow at long time scales.
So this is some sort of like effective diffusivity at the end, basically. So the line that the last line on your left is kind of the effective diffusion due to the crowding.
That's right.
Okay.
So that's normal diffusion. MSD is sort of 1, but it's slow diffusion. So that's diffusion in crowding systems. My sort of interest mostly is a practical diffusion coefficients and what do you use for simulation. And for most things, this anomalous region is not that important, is my sense. What matters is the long-term diffusion rate. And for that, this paper by, I think, off here by Dixon Berkman, they show surprisingly little effect. Well, this is the ratio of cytoplasm to water diffusion. They show for a lot of proteins, this is a factor of 4 slower, essentially, cytoplasm to water. So sure, things will slowly get bigger, but as things get bigger, the ratio from water to cytoplasm is about a factor of 4. Now, things like DNA, that's a long thin thing, obviously, because of entangle and stuff. So DNA has a larger effect as it gets bigger, but more globular stuff has less effect. So in that work and other work as well, these are my rules of thumb. It's a factor of 4 slower, you can have cytoplasms, 15 slower bacteria, 1,000 slower membranes, 4,000 slower bacterial membranes. So that's sort of what I've found based on published papers I've found. So now reaction rates are an interesting question, and very little research on that, it turns out. This is an article by Minton from, say there, I guess like late 90s. Here's binding occupancy, that's how crowded it is. So down here is no crowding, up here is high crowding, and here's reaction rates. He says reaction rates ought to be something like here, just go faster and faster and faster as you get more and more crowding, and then dropping out slower. So you get faster because of volume exclusion, basically your volume, and then slower later on due to slower diffusion. So one exclusion exclusively, diffusion exclusively, and together depends, rise and fall. So based on that, I said, well, what's the, can I do that same thing with simulations? Again, this is an Untitled crowd. This is what we did. This is the group working on us. And the goal was just that primitive simulators and how it worked. A plus B goes to C, A is red, B is blue, black is Crowder's, actually goes to, was it B, sorry. So B concentration is kept fixed the whole time. My hypothesis was this equation. Basically the Collins and Kimball equation, those who know it. Collins and Kimball said, first of all, beforehand, Stochastic equation is K equals 4 pi sigma d. That's diffusion constant. And then if you add in an intrinsic reaction rate, Collins and Kimball say 1 over K equals 1 over 4 pi sigma d. So same thing exactly. Plus now add in a intrinsic reaction rate constant. So that's the Collins Kimball formulation. And they're saying reaction rates are also slowed down by this intrinsic reaction rate constant. So it's the same thing exactly said. I'm going to guess that it's the same thing as this equation here, but add in some volume terms, minus phi there, some volume acceleration. And then diffusion coefficient is diffusion coefficient of the system. So now, was it true or not? Measure diffusion coefficients.
So your 1 minus phi is the excluded volume that's accelerating the reaction.
Correct. Okay. Yeah. So I showed you that it was a very naive hypothesis. I said it's simple, it makes sense. Let's try it out.
And how many dimensions is that? 2.
3D.
3D.
So I'm going to present simulations and first measure diffusion coefficients. So here's V or crowdiness, here's diffusion coefficient, and sure enough goes down as you get more crowding.
That's model, or,
Yeah, the small
this
simulations.
is a small simulation.
Yeah.
Okay.
Yeah, all simulations from small
So the data is from simulation or from experiment or what?
simulation. So not just simulation. Yep. The line is just playing a fit line to it, nothing special.
This is what you get from the small hole formula because you have volume here, this is apparent volume. So this is volume, this is 1 minus V. So you get actually what you have, your guess is actually proved.
Well, no, this is diffusion coefficient. This is not.
This is 1 minus,
Reaction
this is, yeah, but this is the diffusion.
Yeah.
Yeah, so this is with 1 minus phi, right? When you increase
No.
this is, what is phi here?
This is no, it's not reaction rates. This is my diffusion. This is only. This is D of phi. D of phi. This isn't. That's reaction rates 4 pi sigma d. That picture shows D of phi.
That's right, so this is what I'm saying. Phi here represents the fraction of space available, right?
Yeah.
So this is associated to the volume available.
Yeah, that's right.
Yeah, so what I'm saying is that this, your guess is correct. This is what we have proved from the narrow escape theory. Okay, I'll explain.
Yeah, good.
Can you just go back to the previous line? So are you basically plotting this
No.
versus phi? No.
No.
You're plotting.
This is here's
You're
K
just
is
plotting
y.
diffusion coefficient
Here's
versus phi.
K, reaction rate constant.
Okay.
Here, y-axis is diffusion coefficient.
So the diffusion coefficient doesn't decay like 1 minus phi. Yeah, it decays.
Not at all. It depends totally on loss details. So diffusion coefficient is intensively, people see, depends on are powders spherical or are the cubes or the cylinders, are they on lattices or off lattice?
And then what is the straight line? So the dots are for simulation. What is the straight line?
Just a thin line.
Okay.
Yeah.
Okay.
It's not theory, it's just kind of pure
Okay.
fact.
Okay.
The diffusing molecules have a volume.
Yeah, yes and no. No, they don't. There's particles here. I expanded the Crowders so we would have a volume effectively. So I placed in their Crowders, and here's my Crowders. I want to have my tracer to be the same size. To do that, I said, make it a dot, expand this So now this dot can't go inside this dashed line, just as though this thing had volume.
And what happens if now the crowders touch each other?
So now they can touch, they couldn't touch that before, now they can touch up to that dashed line.
Yeah, so
So-
that's what I'm saying. What happens if the dashed lines touch each other?
That affects things.
Okay.
Yeah, because,
So that basically means that the molecule cannot go through that.
that's right. Yeah, and again, this curve here depends on everything possible.
Okay.
This means not interesting because it depends on everything possible, it depends on shapes, overlaps, structure, yes.
Okay.
But it's just a curve. Here's the interesting part, is reaction rate. Here's number of A molecules as a function of time. So A molecules start high, decreases. Find the slope of this curve, that's reaction rate. And here's a slope. And reaction rate starts fast, and then slows down to a steady state rate. And it gets more and more noisy as you get fewer and fewer molecules. So now, fit this curve with this function. This is from Stochastic theory. Let's say what's our steady state long-term reaction rate based on this fit or reaction rate data. Sort of, what's this thing approaching as reaction rate curve? Reaction rate. From that, here are my results. Dots are simulation results. The blue line is my initial theory. I was wildly off. And then red line, it fits. It's the same thing, but I added a gamma function coefficient here. So my function does fit if you add in a gamma parameter. Now here's the diffusion limited part. Here's the admixture limited part. And it looks nice.
So John Chapman and Maria Bruna have done the asymptotics for diffusion with the hard sphere crowders.
Okay.
And they derive effective equations and they get what the scalings and the exponents are in that exact case.
Mhm.
So you could look at how do your simulations match up for the predicted scalings
Yeah.
they're getting. So I don't think they've done reaction in that case, but that essentially is going to give you kind of the volume fraction behavior.
Let's see.
And so, and here D, in principle, is not a function of phi.
It is. I use
No,
this.
in the formula, when you
It
have.
is. Yes, it is. D is function of phi. It should be in there. Yeah, I should have said d of phi in here. Sorry.
Okay.
Yeah. So I use this d of phi function.
Okay.
And I plug that into.
Yeah, just to make sure that I understand. That's fine.
Yeah.
Thank you.
Yeah, same thing here. So that's actually to be d of phi there. And so, end result, I get a nice fit to this modified hypothesis, but it's wildly off from the original hypothesis. That's for admixture reactions. I now do diffusion reactions. Again, here are dots. Here's my theory, my hypothesis. And here's a modified hypothesis. Now a different gamma value. So, I don't know. So conclusion is, oops, I was wrong. Mitten was wrong too. That's what I was comparing to was the original work. And I have a curve that fits, but I don't know why. So, open questions. What's going on here? So can I actually understand this and figure out what's going on? What else doesn't fit?
If you look at mean first passage time and the distribution of arrival time from one spot to another spot.
No.
This is, you look at the distribution. So I expect, depending on how crowded is the environment, and if you have small passages, because you see when you start doing crowding, you have these small passages
Yeah.
that are the bottleneck. If you have at least some of them, then the arrival time should be Poissonian distributed more or less.
Yeah. I have a guess actually. My guess is different from that, but at least we'll look into certainly. My guess is diffusion coefficient is taken care of adequately with that d of phi thing. There's diffusion.
Yeah, because I mean,
Yeah.
this is what we observe. I mean, when you
Yeah.
get this you get when you have enough crowdedness, you get exactly this shape. So this I think you have captured.
Yeah.
So, but for, getting information about this arrival time from, let's say, you start somewhere, you ended up some small part.
Yeah.
This would be interesting to look at the distribution, and then when you get this exponential, I think it might be Poissonian, to link this Poissonian and to look at on the way of all the particles that arrive here that contribute, if they go through domains with narrow passages And what are these passages that are of interest? That they contribute. The rest isn't important.
Yeah, could be.
And then okay.
If I look at that some more.
Okay, we can talk about this.
Yeah, there's nothing unfinished work here. That's what I'm presenting it here is to be done. So I'd love to work with you guys. My hypothesis is different. My hypothesis is that if you have a surface here, a polyline surface, put an A molecule right here and fix it there, then B molecules are diffusing in and reacting with it. This surface proximity slows down reactions because it's there. If things farther away, there's less effect. If it's closer in, there's more effect. So surface proximity actually slows down reaction rates. So my guess here is that Crowders are a whole lot of surfaces that having effect on those reaction rates. But I'm not sure. It's my guess. So let me end up here.
So was Minton's measured or just hypothesized?
Sorry?
Was Minton's plot hypothesized or
Hypothesized. He explained and said, this is my guess, and published it. That's been the definitive word for the last 20 years. A sort of goal of our conference was to compare these simulators. Small but not great. Space slightly impressed me actually, it's really fast and less artifacts. And ML space was promoted, so it didn't work that well. Kappa worked, was impressive in itself, because Kappa is a non-spatial simulator, so that it worked well was amazing. GFRD was really slow. GFRD, what took me 10 minutes, took them 4 days. So it wasn't actually useful in this case. Questions? How should I answer questions? So I'm going to stop here, but explain. Before I talk about more, I'll talk about information transfer in biology. I would talk about signaling in cell systems from input to output. I'll skip this though. I'll mention a couple of questions though. So the big question here is cell signaling systems transfer information, and they're known pretty well how they work. We know that cells lead by receptors then transmit signals to something else, and so forth and so on. This whole signaling pathway. It's often known in pretty good detail what happens. What people do not know is how much information is there in the pathway. They say, well, here's the pathway, but is it noisy or is it not noisy? We just don't know. And some work here suggests it's noisy. Obviously, I think it's actually not noisy, but it's I think, a major question to be addressed. And so, open questions. Questions like calculate number of bits transmitted through information signaling pathways, and then once you have that, what information lost in that pathway? What possible put into other information? For example, there's other pathways in this cell, and there's information, and there's one pathway exclusively, or is it sort of spread out to other pathways too? So sort of what's the crosstalk of pathways? So you can actually quantify that, not in sort of fuzzy way, let's say, how many bits get from here to here. And then what some mechanisms they would go to bad information transfer. So how cells evolve to naturally. And then thanks to lots of people here and elsewhere.
For the reactions and the crowding simulations, you're doing the Collins-Kimball model. So you're using the tweaked version of Smolden? Or did you just expand out your reaction radii? I mean, how is in the base version, right, you don't have reflection.
I think it's time step. You just change time steps.
You just changed your time step.
I believe so.
So you didn't actually in the simulation put in the possibility of non-reactive.
That's right. I think it's changed time steps. So a long time step that does more activation limited. Because reflection off the surfaces is basically exact, even though it's long time steps. So you can do multiple reflections in a single time step.